Glycolysis. First of all, what's the meaning of glycolysis? Glyco means glucose and lysis means breakdown. So the word glycolysis means the breakdown of glucose. Now its definition. The stepwise enzymatic breakdown of glucose into two molecules of pyruvic acid with net gain of 2 ATPs and 2 NADH2 is called glycolysis. So now it's the complete definition of glycolysis. The first thing is stepwise. Stepwise means a series of reactions. So in case of glycolysis, there are a series of reactions, and each reaction, each step requires an enzyme. And glucose, which is actually a 6 carbon compound, it is broken down into pyruvic acid, into two molecules of pyruvic acid, and each pyruvic acid is actually a 3 carbon compound. So at the end of glycolysis, a single glucose molecule is broken down into two molecules of pyruvic acid with net production of 2 ATPs and 2 NADH2. And next is the site of glycolysis. Site means where glycolysis occurs. Glycolysis is basically the anaerobic phase of respiration. It does not require molecular oxygen, and it occurs in the cytoplasm of the cell. And next, the phases of glycolysis. Glycolysis can be divided into two phases. The first one is preparatory phase and the second one is oxidative phase. The first one, preparatory phase, means that it is the energy investment stage. In this phase, the phosphorylation of glucose molecule takes place by the consumption of 2 ATP molecules from the cytoplasm. So in the first stage, which is the energy investment stage, in this case the glucose molecule uses 2 ATPs from the cytoplasm. Second is the oxidative phase. Oxidative phase is also known as energy harvesting phase or energy producing phase. In this phase, the energy is released from glucose, and this energy is used in the production of 4 ATP molecules. So in the first phase, glucose uses 2 ATPs from the cytoplasm, and in the next phase, glucose molecule produces 4 ATPs. So there is the net production of 2 ATPs. Now I am showing the steps of glycolysis. So in the first step, the glucose, which is a 6 carbon compound, it is converted into glucose 6 phosphate, and the enzyme used here is hexokinase. So the glucose molecule utilizes ATP from the cytoplasm, and the inorganic phosphate attaches with the carbon number 6. So glucose is converting into glucose 6 phosphate. On the right side of the board, you can see the linear structure of a glucose molecule. So glucose 6 phosphate means that on the carbon number 6, the phosphate group will attached, which is now I am showing. Now you can see glucose 6 phosphate. In the next step, glucose 6 phosphate is converting into fructose 6 phosphate. In the next step, glucose 6 phosphate is converting into fructose 6 phosphate. Actually, glucose and fructose are the isomers of each other. They have the same molecular formula but different structural formula. So the enzyme required is, here is a isomerase enzyme, and its name is phosphoglucoisomerase. And on the right side of the board, you can see the fructose 6 phosphate. In the next step, Fructose 6 phosphate is again utilizing an ATP from the cytoplasm, and ATP is converting into ADP and inorganic phosphate. The phosphate comes to attach with the carbon number 1 of fructose. So as a result, fructose 1,6 biphosphate is formed, or you can say fructose 1,6 diphosphate. And the structure of fructose 1,6 biphosphate is shown on the right side of the board. And this fructose 1,6 biphosphate is also a 6 carbon compound, and the enzyme required here is phosphofructokinase. In the next step, fructose 1,6 biphosphate is broken down into two molecules with the help of an enzyme which is known as aldolase. And two molecules are produced. Each molecule is actually a 3 carbon molecule, and both molecules contain a single phosphate group also. So the first molecule is dihydroxyacetone phosphate, DAP, and the second molecule is known as phosphoglyceraldehyde, PGAL. So both these molecules, they are 3 carbon compounds, and both of them contain a single phosphate group. Up to here it was the first phase of glycolysis, in which energy is required and energy is taken from the ATP molecules. DAP and PGAL both are the isomers of each other, and both these molecules, they are interconvertible. The enzyme here is triose phosphate isomerase. So with the help of this triose phosphate isomerase enzyme, and then in the next step the DAP will be converting into PGAL. So now we have two molecules of PGAL, which is actually phosphoglyceraldehyde. In the next step, there are two molecules of phosphoglyceraldehyde. So the phosphoglyceraldehyde utilizes a phosphate group, inorganic phosphate from the cytoplasm, and it is converting into biphosphoglyceric acid, which is again a 3 carbon compound. And the NAD, which is a coenzyme, NAD is reduced to NADH2 with the help of a dehydrogenase enzyme. And you can see I have written 2 with phosphoglyceraldehyde and 2 with the IP. IP means inorganic phosphate, and 2 coenzymes NAD are reduced to NADH2, and as a result the 2 molecules of biphosphoglyceric acid are formed. And biphosphoglyceric acid is again a 3 carbon compound. In the next step, with the help of an enzyme phosphoglycerate kinase, the biphosphoglyceric acid gives its one of the phosphate group to ADP and results in the formation of ATP. And the biphosphoglyceric acid converts into a phosphoglyceric acid. In the biphosphoglyceric acid, there are two phosphate molecules, while in the phosphoglyceric acid, a single phosphate group is present. And the second phosphate group is having with the ADP, and it's now converted into ATP. So it's the first step in which the energy is produced. It's the energy harvesting step, and 2 ADP, they are converted into 2 ATP. So here 2 ATPs are formed. In the next step, phosphoglyceric acid PGA loses a water molecule, and with the help of enolase enzyme, it's converting into phosphoenol pyruvic acid. In the last step, The phosphoenol pyruvic acid loses its phosphate group, and with the help of pyruvic kinase enzyme, it is converted into pyruvic acid, which is a 3-carbon compound. So phosphoenol pyruvic acid loses its phosphate group, which is gained by ADP, the adenosine diphosphate, and ADP is now converted into ATP, which is energy rich molecule, adenosine triphosphate. So now you can see this is a complete um, diagram of glycolysis, and it's a stepwise process. You can see there are about 8-9 steps. And enzymatic means almost every step needs an enzyme, and a glucose, a 6-carbon compound, is converted into 2 molecules of pyruvic acid. You can see at the start is a glucose molecule, a 6-carbon compound, and at the end there are 2 molecules of pyruvic acid, which is actually a 3-carbon compound, and with the net production of 2 ATPs and 2 NADH2. Now I am going to explain the net ATP production during glycolysis. In the glycolysis, as a whole, total 4 ATPs are produced, and you can see here it on the left side of the board. And in the start of glycolysis, 2 ATPs are consumed in the first phase. So the net ATP means we will minus the 2 ATPs from the 4. So 4 minus 2 equals to 2. So the net result of ATP formation in glycolysis is 2. So glycolysis means one glucose molecule is converted into 2 molecules of pyruvic acid with production of 2 NADH2 and 2 ATP molecules, and it occurs in the cytoplasm of the cell. So it was all about glycolysis. Thanks for watching the video, and don't forget to subscribe the channel.